Hi! Today we're going to learn about um, mitosis and cytokinesis. Chromosomes condense at the start of mitosis. A chromosome is a one long continuous thread of DNA that consists of numerous genes along with regulatory information. Our body, the human body, has 46 chromosomes each. The DNA wraps around proteins that helps organize and condenses it. In interphase, the DNA is loosely organized and looks like spaghetti. Specific genes are accessed to make proteins and copy entire DNA sequence. In mitosis, chromosomes are condensed. Duplicated DNA must be condensed to divide two nuclei. These changes in the DNA makes functions possible. So the key point here are, are the DNA and proteins. Chromosomes structure. We have the DNA double helix. The DNA double helix. Each continuous stranded DNA molecule makes one chromosome. Next, we have the histone or histone. I'm not really sure. It is a group of proteins. DNA wraps at regular intervals around the proteins. Then we have the chromatin. It is a loose combination of DNA and proteins. Then, last, we have the supercoiled DNA. Its chromatins coils more and more around organized cells. Here, in the bottom right, we can see a chromosome. One half of a duplicated chromosome is a chromatid. Sister chromatids are held together at the centromere. Telomeres protect the DNA and do not include genes. Here, we can see a chromosome which is condensed. Here in the half, in the square, we have a chromatid. Two of them makes sister chromatids. In the center, where the chromatids intersect, we have the centromere. And the telomeres are made of repeating nucleotides that doesn't form genes. The telomere prevents from the chromosome from attaching to others, and some codes for new, and some are lost for new. Sorry. Mitosis and cytokinesis produce two genetically identical daughter cells. Interphase prepares the cell to divide, and during interphase, the DNA is duplicated. Here we can see a parent cell with a nucleus, with the DNA, centrioles, spindle fibers, and centrosomes. First, we have the prophase. It is the first stage of mitosis. Chromatin condenses tightly into coiled chromosomes. The nuclear envelope breaks down. The centrioles begin to move to opposite poles and form organized microtubules. Spindle fibers go from centrioles and radiates toward the cell center. Then we have the metaphase. Spindle fibers attach to each chromosomes and aligns in the cell equator. Well, the equator is the center of the cell, like the middle, yeah? Then we have the anaphase. Chromatids separate from each other to opposite sides of the cell. Cytokinesis forms in late anaphase or telophase. During the telophase, nuclear membranes start to form. Chromosomes begin to uncoil and spindle fibers pull apart. There are some differences between animal cells and plant cells. In animal cells, we have the cell membrane. The membrane pinches closed, while plant cells, cell plate forms. And that's it. Oh, and cytokinesis. It it divides cytoplasm between two daughter cells, each with a genetically identical nucleus, and the cells enter interphase and the cell cycle begins again. And that's it.